Well, thank you very much. And now we will go over to the last presentation of this morning. I would like to invite Dr. Marshall from Australia to discuss the microbiome, which feeds the myriad of autoimmune diseases. Dr. Marshall.
Thank you, Mr. Chairman. And now for something completely different.
So a very provocative title. We're talking about the microbiome, the microbes which inhabit or cohabit our human body, which feeds a myriad of autoimmune diseases. Which feeds, causality is implied there. Can I support that? And a myriad of autoimmune diseases, what are we talking about there? Well, let's look back a little bit in history first. At each of the last several congresses, my colleagues, my clinical collaborators, have given clinical results showing how sections of our cohort or case series, and in some cases case studies, have been responding to the therapy that we developed in order to counter the effects of the microbiome. This is from Porto, and Captain Tom Perez, who had just retired after 25 years at the FDA, presented a small retrospective of our cohort. In this case, about 103 patients, if I remember correctly, with conditions ranging from rheumatoid arthritis through to myasthenia gravis and diabetes insipidus through psoriatic arthritis. You can see there are more and more patients as we go up towards rheumatoid arthritis, Hashimoto's, uveitis, psoriasis, the more common conditions, with a detailed status of how the patients were getting along. Most of them were resolving quite nicely. Also in Porto, Dr. Greg Blaney from Canada reviewed a couple of particularly difficult cases that he'd been working with. This one was an MS patient who started with an EDSS of 8.5, was wheelchair bound and incontinent, and through to 2008 had improved the EDSS, was no longer incontinent, and no longer needs diapers, in fact, and to this day that patient is continuing to do well. And then again in Ljubljana, 2010, Dr. Greg Blaney gave another couple of case histories, discussed another couple of case histories. In Singapore, at the Asian Congress last September, Dr. Goetz Sepelka, a psychiatrist from Germany, was talking specifically about how psychiatric manifestations, particularly depression, manic depression, and to some extent schizophrenia, were responding at the same time that the inflammation, the chronic inflammation, was resolving. And then just a few days ago, Inga Linseth from Norway, collaborator from Norway, gave a case series of 63 subjects who, both Canadian and European, who were demonstrating resolution of chronic fatigue. Chronic fatigue being, of course, one of the most difficult diagnoses that we've found to deal with. So what's this all about? Well, let's go back in time even further. The discussion on infection and autoimmunity came up on the Autoimmunity Network, Yehuda's Autoimmunity Network, and those of you who aren't members, please join and engage in the discussions there online. And one of the comments that was made was, this has been a hot topic for decades. Yes, indeed. And in fact, if we go back 3.5 decades, we'll find that the father of this family of autoimmunity, Yehuda himself, was already looking at the links between infection and autoimmune disease. And I think by a couple of years of that, after that, Yehuda had identified MTB as being one of the potentially key pathogens. And at that time, I was just fooling around with insulin infusers and infertility, but never mind. But you know, something's changed in the last decade. Something very important happened a decade ago. And that was, we got a complete working transcript of the human genome. Now, I have to admit that nothing much has come out of that yet in terms of direct genetics. But another field that sprang up from the field of genetics, the field of metagenomics, is turning medicine on its head, turning medical knowledge on its head. It'll take several decades to turn medicine on its head, turning medical knowledge on its head. And this burst of discovery is the microbiome, the human microbiome. Now, NIH initiated a project in 2007, and it wound up late last year, earlier this year, called the Human Microbiome Project. And this was a project which was to define exactly what microbes were associated with Homo sapiens and to try and figure out what they were doing in and on Homo sapiens. And what was concluded by the project was man is a superorganism. We all carry microbes. We carry microbes not only on our external surfaces, but also in our bloodstream, in our tissues, in our cells. And millions of genes belonging to thousands of species of microbes interact with the 25,000 genes of the human body. And that interaction gives the potential for dysfunction. The microbes can cause the human body to dysfunction in extremely complex ways, because you're dealing with 25,000 genes being worked on by millions of, potentially millions of microbiome genes. And then, of course, you've also got the viral genes and the fungal genes, because they're also persistent funguses. And one of the conclusions that came from the Microbiome Project is that it is our microbes which make us genetically unique. Every one of us has a different microbiome to the person sitting next door. Every one of us has a unique microbiome. It varies between people, and it varies from month to month, and in some people from week to week. It varies when we travel to a different region of the world. The microbiome changes. Metagenomics is the study of the genetics of how those microbes interact with the human body. And this book was written by Karen Nelson, or edited by Karen Nelson, who was really the instigator behind the HMP. We wrote the chapter in there on autoimmunity, and it goes into a lot more detail, I think about 35 or 40 pages, that I'm going to go into in this talk. But if you want to find out more about exactly what the microbes do and how the microbes make us genetically unique, then that is the source for you to go to. But here's a quick summary. Firstly, babies are born dirty. They already have a gut full of microbes. But in addition to a gut full of microbes, they also have a body full of microbes. They bring many of the microbes of the mother, some of the microbes of the father, and within a few weeks after birth, they have accumulated most of the microbes of the extended family. I guess grandmother has to kiss the baby. This is the key start to our life. We start our lives with the maternal heritage, with the maternal line. So you can already see, those of you that have been studying autoimmune clustering in families can start to see now how this occurs. So we start off with the microbiome. Then we can't avoid it from that point on. From that point on, we... sorry, the screen here says 2 minutes left. From that point on, the air is full of bacteria. And this is the air in San Diego Indoor Hospital. This is from the J. Craig Venter Institute. Some work done by Craig Venter, the first person to get a full human genome transcript, and one of the leaders of the metagenomics initiative. And you can see in a hospital, okay, bacteria, 86% of the DNA in the air. That's not really surprising. But look, you go into a house and it's almost identical. The human DNA in the house is a little bit less dense than it is in the hospital, but we get exposed to microbes, microbial DNA, and also fungi and insects in the air. Can't get away from it there. We get it in our food. This is a wonderful study that was done by Eric Elm's group at MIT. And what they did was look at the microbes in the human body, just up in this region of the chart, and plot a heat map for the correlation between those microbes in the human body and microbes from other sources. For example, farm food sources here, and wheat and other agrarian food sources here, and soil and hematroph, hydrothermal, etc. But the thing that's really interesting is this particular section of the heat map is a plot of all the genes which are thought to confer antibiotic resistance. In other words, there are certain genes in microbes which which science has identified as being associated with conferring antibiotic resistance when they're transferred from one host to another. And you can see that those genes that cluster in man also cluster very heavily in the farm and the food categories. In other words, we get our bugs, and particularly the genes that we least want, from our food. That's, of course, exacerbated by today's modern international transport of food, and also international transport of human beings, including ourselves. So where do these microbes cause the most trouble? They cause trouble when their genes can interfere with the transcription of the human genome. When they get inside the nucleated cells of the human body, that's when they cause trouble. This is a slide from 2.5 decades ago from the Warosco group at Columbia University, who did a study of several diseases, Crohn's, juvenile RA, and sarcoidosis, looking at lymphocytes and macrophages, monocytes, specifically for infections, nucleated staining within the cytoplasm. And this JRA lymphocyte, you can see there is clearly a vacuole here, which has not undergone phagocytosis, and quite a reasonable clump of DNA there. There are also DNA lining these longer tube-like, well, they could also be plate-like because we don't have 3D objects as well. When the microbes can persist in the macrophages and the monocytes of the immune system, then as far as the Homo sapiens is concerned, it's game over. And indeed, that is what seems to be happening in chronic disease. Somebody said to me the other day, all you're seeing in blood, because anybody that's got the capability of measuring the microbes with the metagenomics tools can look in human blood and they'll find many, many microbial DNA samples from that blood. Human blood is not sterile. Human tissue is not sterile. But of course, most of the microbes can't be cultivated, which is why we haven't seen them before. Somebody said to me, but all you're seeing are DNA fragments, not real organisms, not live organisms. Well, that's a fascinating criticism because, firstly, you'd have to have a pretty weak immune system if that were to allow DNA fragments of microbes to be floating around in peripheral blood. And secondly, these DNA observations merely bolster the observations of, for example, Warosco's on the previous slide, and many others that have gone before. Indeed, you yourself can observe it's moving now. You yourself can observe these infected cells in whole blood if you use the correct processing and make sure you're looking at very fresh blood, because these cells, monocytes, lymphocytes, very, very quickly disintegrate following removal from the bloodstream. And that's another reason you don't see them very much in vitro. All you see is fragments left. The microbes get out of the... I wonder if I can go back. The microbes get out of the infected cytoplasm very quickly with these long fili, characteristic of microbial migration in blood. So it was pretty obvious to me back a decade or so ago that we needed a different type of microscope. And at that stage, groups at the University of Illinois and also in Europe were looking at the possibility of doing computational microscopes. In other words, microscopes that are capable of examining 3D structures at the level of molecules. And in fact, these microscopes are made of chemistry, physics, math, and software. Oh, and of course, supercomputers. Basically, what the software does is emulate every force, every location of every atom in a molecule, thousands at a time, in order to try and figure out how the molecules interact and how the molecules stay in homeostasis. And that tool allowed us to look at molecular mimicry. And what we found with molecular mimicry was something quite different from the concepts that have sprung up by observing it in connection with antibodies. Firstly, what we have on the screen are two proteins. They are transmembrane proteins, GPCRs. This one is a human angiotensin II type 1 receptor. It has a drug in the binding pocket. And this one is a protein called YDGG from E. coli. It is one of the many, many proteins in E. coli, and it happens to be essentially identical with a human angiotensin II receptor. It quite easily would embed itself in the cell wall. Heaven knows what it would do, and heaven knows what the cell's mechanisms, the Golgi mechanisms, etc., would do when faced with this type of protein in the cytoplasm of a cell. But you can see it's identical. The shape is identical. I only have helixes here rather than each individual atom, of course, because there are thousands of atoms on each of those receptors. But you can see it's identical. There are slight differences. You can see the drug binds at a different spot. Well, wait a minute. The drug binds into a bacterial protein? Drugs affect the microbes? Wow, that's an interesting observation in and of itself. So, working on the basis of a better understanding of what microbes are and what they do, I mean, many of these proteins are needed for the microbe to work. E. coli, for example, produces energy from glucose. And it produces energy from glucose with almost exactly the same intermediate products as Homo sapiens does. So when you look at the genes associated with that energy conversion, you find they're almost identical to Homo sapiens. You put those into a mix in the cytoplasm of a cell, you're going to get dysfunction. And another key thing we found by using the computer microscope was that the key innate immune functions are unique to Homo sapiens. There is one weakness in Homo sapiens' innate immunity which doesn't exist in any of the other mammals, including the higher primates. And that is that one receptor is responsible for cathelicidin, LL37, the key intercellular microbial defense, beta-defensins, and TLR2. TLR2 is a key intracytoplasmic toll-like receptor. And these are essential to the intracellular innate immune defenses. And in Homo sapiens, and in only Homo sapiens, there is one nuclear receptor that is key to expression of all those key innate immune functions, and it is the VDR nuclear receptor. And in order to persist inside cells, microbes have to knock out the VDR so they don't have to deal with the cell's defenses. And in fact, that's exactly what they do. Let's take a classic microbe for chronic disease, EBV. EBV knocks out VDR very, very effectively by about 15 times in magnitude, looking at the lymphoblastoid cell lines after 1.5 years, for example. Peripheral blood cells, not a big deal. but by the time you get through to the lymphoblastoid cell lines at 2 months and 1.5 years, EBV is really knocking down the expression of the human proteins, and particularly the VDR. Other microbes that are already known to also knock out the VDR function, Mycobacterium tuberculosis, Borrelia burgdorferi, Chlamydia trachomatis, Aspergillus, HCV and CMV. Reads like a rogues gallery of the microbes that we see in autoimmune disease. But you know, the microscope can do other things as well. When I do highlight individual atoms within the VDR, this is a VDR, when I do highlight individual atoms, you can actually see how the receptor activates, because these two entities have to bind to the DRIP205, which sits across here, to form the heterodimer. Well, the RXR heterodimer, and therefore transcribe genes. And when we place a ligand in the binding pocket, it forces these atoms apart so they don't bind and they can then take the coactivator. And with no ligand or with an antagonist in the binding pocket, that doesn't happen. So we were able to study activation of receptors as well. And what we found was that there is a molecule which is effective, very high affinity for the binding pocket of the VDR, and which very effectively can overcome the effect of the microbes on the VDR. And that is a molecule called olmesartan, which is a sartan licensed for blood pressure, hypertensive indications. The main thing we have to do in retargeting it is to make sure that the concentration remains reasonably constant by giving a pulsatile dosing rather than the normal daily dosing of the olmesartan you see in green. By giving a more frequent dosing, we manage to keep a basal level in the bloodstream. We need to do that because a nuclear receptor lifespan is only hours, 3 to 6 hours for the VDR, and then they've all turned over. So you've got to keep the concentration in there. That is the secret of retargeting. But look, that's really old technology. My thesis was full of that. Well, at this point, having gotten the fundamental understanding of what was going on between the microbes and mankind, we were able to start focusing on patient-important outcomes. And we were able to skip the phase of mouse-important outcomes. So whereas with mouse studies, I shouldn't say no loose ends, but you try and get no loose ends, certainly few loose ends. In our case with patients, you have a lot of loose ends. You have to ask whether those reports that I showed you at the start of the presentation are perhaps just placebo effect. Are they perhaps just outliers, outlying responders, or are they indicative of something more definite, more important? So, how does the microbiome actually cause disease? Well, it messes with our human body because inside each cell, many human metabolites are affected by the genes. And you know, you might have some genes from bug A combining with the genes from bug B, which might lead to a body dysfunction X, which is just one syndrome that would add up to a disease diagnosis. Or you might need genes from bug C, D, E, and F with the absence of genes from another pathogen, because many microbes are pathogenic to each other. Strep and staph are two examples. The genomes accumulate gradually during life, and it's genes from the accumulated metagenome which determine the clinical dysfunction and the disease symptoms. And that is why this comorbidity chart shows so much interrelationship between the various autoimmune and other chronic diagnoses. If we plot a chart of the top 120 diagnoses in the cohort we're following, starting at CFS, sarcoidosis, fibromyalgia, hypothyroidism, RA, going with many cases in that, going through to PTSD, epilepsy, sciatica, seizures, etc. That is a pretty big cross-section. And in many cases, I've just lost my slide here, don't worry. In many cases, you'll see that, those are the types of diseases that you see.
Uh-oh, I've lost my presentation. Can you get me back my presentation? I can probably do it. If I go down here to here, and then I shift F5. Am I back? Yes, excellent. Okay.
So the key thing is that when you're dealing with microbes, they have two effects. The first effect is inflammation. That's the body's response. But the second effect is the molecular mimicry. And they create different symptoms in the patients. From the inflammation, you're going to get the cytokine storm, autoantibodies, nitric oxide, reactive oxygen species, etc. From the molecular mimicry, you're going to get the interactome damage, the interactions, which leads to body dysfunction, cortisol axis dysfunction, thyroid axis dysfunction, etc. In a way, you can look at these persistent antiphagocytic infections as something like white ants. There's no obvious sign of damage while they accumulate until the whole structure starts to crumble.
Okay, there is a problem. The problem is immunopathology. I am summarising now. This is my last slide, in fact.
There is a problem. Immunopathology. And what you have with immunopathology is you have direct damage by the pathogen on the host. Then you have the host resistance mechanism, including the inflammation, back on the pathogen. But some of those mechanisms also cause damage to the host itself. Immunopathology. And that is a big problem, because once we re-enable the innate immune system so that it can see these intracellular microbes and starts to kill them, then the level of inflammation rises, the level of symptoms rise, and that's a problem when we're dealing with people who are right on the edge in terms of being able to manage from day to day in the first instance. We recently published a paper, Immunostimulation in the Era of the Metagenome, which specifically deals with immunopathology for those of you who are interested. So thank you, Mr. Chairman.
Thank you very much for this new topic and stimulating lecture. Time for one question. Yes, please.
So, just very quickly, the BDR is the vitamin D receptor, and you're suggesting olmesartan as a partial agonist. Vitamin D is a very good agonist of the vitamin D receptor. Why use olmesartan?
Because vitamin D has to be used at such a high concentration that it wipes out other receptors as well. We published a paper on that a couple of years ago in one of Yehuda's summaries. So it knocks out thyroid, it knocks out cortisol, mineralocorticoid receptor at the concentrations that would be needed to get the BDR working again. The 125D has to go too high.
Okay, well, thank you very much. I think it's time to talk to the speakers and the audience for their attention.